Hi, I'm Jay Phelan. Let's talk about some biology. Today we're going to talk about what is Mendel's law of segregation. Genetics, the study of heredity, concerns itself primarily with why is it that offspring look more like their parents than some other random individual in a population. And it's a complex, rich, huge field. However, to truly understand eukaryotic genetics, it all boils down to one important idea that Gregor Mendel had a long time ago. Everybody knows a few little facts about Mendel, right? He was a monk. He lived in the mid-1800s. He liked peas. This can tend to give us the wrong perception of this lonely old guy off in isolation with 4 pea plants on his windowsill and trying to figure some stuff out. What Mendel was doing, we have to think about in the context of what was going on in the world of scientific thinking at the time. He was in the Moravia, which is now the Czech Republic, working in a monastery. But monasteries were real hubs of scientific research. And at the time, Charles Darwin had just published The Origin of Species in 1859. And this rocked the world. What Darwin had laid out was a mechanism by which species could change over time. And this rocked the world because people had been working with the assumption that extinction never occurred and that species were all put here at the same time and they never changed. So if they could change, that was pretty radical, including because it had some implications for humans. Maybe we weren't put here in this form, so what did we change from, and so on. So this was the world in which Mendel was doing his work. So Darwin's book had come out 1859, and in it, he laid out a mechanism. The mechanism was very straightforward. He said, if you have variation for a trait, this could be how much milk cows produce or how tall people are, if there is heritability for that trait, in other words, offspring resemble their parents more than some random individual for that trait, and if there's differential reproductive success based on that trait. In other words, if you're a dairy farmer, you only reproduce the cows that produce the most milk. If those 3 conditions are satisfied, then you have evolution by natural selection, and your populations can change over time. What's weird, though, is that Darwin laid out all sorts of information about variation, several chapters on variation and on differential reproductive success. When it came to heritability, though, he said, we don't really know. All you have to do is look at your friends. Their kids look like the parents. So we know it's true. Or animal breeders. They know that it's true, but he didn't know how it worked, so he kind of left it at that. And that's where Mendel comes in. So Mendel was working in this monastery. When he first got there, he had been educated in Vienna, and he was very excited to have some kind of research career there. And you can still see the monastery where he did his work. And what they would do there is the head of the monastery, when Mendel got there, was a guy named Abbot Knapp, the guy in the front row with the cross around his neck. He would assign people to various tasks. And he assigned Mendel to study heredity, because this was a big issue in the world. Mendel, in this picture, you see him in the back row, second from the right. They forced him to hold a little pea plant. But anyway, he also was going to be a teacher. They would support themselves by teaching at local high schools. Mendel wanted to teach biology. I failed the test 2 times, and so never actually taught high school biology. He taught physics, high school physics, throughout much of his career while he was there. But he was active in the world. He went to meetings throughout Europe. You see him popping up at these various conferences. He was thinking about stuff, and he was talking with the leading researchers of the day. So Mendel wanted to figure out heritability. How does it work? We know it's true, but how does it work? And he worked using pea plants, and as we'll see in a minute, that's kind of irrelevant. But what he had to do was look at parents. What do they look like? What traits do they have? And what do their offspring look like? And just keep doing that over and over for a lot of different traits with very large numbers of organisms. He didn't have 4 pea plants in his windowsill. He was looking at sample sizes in the thousands, more than 10,000 overall. So he did this for several years, and he finally was ready to unleash this on the world. So he gave a talk at this place called the Natural History Society of Brünn. And like many biology organizations, they have this big logbook. It looks like one of these Harry Potter books. You can crank it open. And they still have the book today. And it'll say, here are the people who came to this month's meeting. Here's what we talked about and so on. And in the logbook, it says from February 1865, it says, Gregor Mendel spoke on inheritance in pea plants. So lots of people came, and then it said this: there were no questions. And this has to rank up there among just the saddest things ever. There were no questions. The whole world is talking about Darwin's theory: variation for a trait, heritability, differential reproductive success. And Mendel gives this talk where he lays out, here's how heritability works, and it's correct. If you look at his paper today, even 150 years later, it looks pretty much like a basic genetics textbook. He lays it out. Here's what's going on. But the world wasn't ready for it. They couldn't quite figure out how to integrate it with what they already knew. So he did more research. He did more and more complex experiments. Eventually, he was taking up a lot of resources, and it wasn't clear to anyone that he was doing something as important as it was. He got bumped up into the administration. They made him abbot, and eventually he had to leave his experiments behind. And it became unclear to him. For a long time, he thought, no, someday people will appreciate it. But then he wasn't sure. He thought, well, maybe I didn't discover anything. So that was kind of sad, and it's all made all the more sad. This is a picture of Charles Darwin's study. And Darwin, probably the one person on Earth who was in the headspace to appreciate what Mendel had done. Mendel sent him a copy of his paper. So after he gave his talk, he published it, sent it to Darwin, and they found it on Darwin's desk unopened after he died. So Darwin got so much mail, he just never was able to catch up and see it. So, the world could have been different if that had happened. I think that's it right on the corner of the desk there. Anyway, let's talk about what it is. In order to appreciate how heritability works, how genetics works, let's talk about all the genetic material that an organism has. We call it its genome. You'll also hear the word genome used to discuss all the genes for a species. But we're talking about for just an individual. In the case of a human, all of our genetic material is divided into 46 linear strands, chromosomes, and they're arranged into 23 pairs. They're arranged into 23 pairs because we get one copy of chromosome number 1 from our mother, one copy from our father. We get one copy of chromosome number 2 from our mother, one copy of chromosome 2 from our father, and so on. So if you were to look at a generic cell in an organism that only had 6 chromosomes, 3 pairs, it might look something like this. So you see 6 linear strands. There's a blue one, there's the green ones, there's the red squiggly ones, and there are 2 copies of each, the maternal set, the paternal set. So we call those a homologous pair. They are going to have the same set of genetic information, the same trait, eye color, hair color, but maybe not the same version of them. You can look for any individual at all of their chromosomes laid out. We call it a karyotype, and it's just a picture of all the chromosomes in a cell. In a human, then, that would be all of these 46 linear strands of genetic material that are floating around lined up so that we could look at them. Now, why would you want to do that? Would anyone want to do that, organize them this way? Yeah, people pay money for a karyotype. I did once, so that you can see all the chromosomes. Why would you want to do this? Well, people do it when they're going to have a baby. They can look at cells that have been sloughed off by a developing fetus, get some of those cells and look at them, make sure that all the chromosomes are there, or look to see, is there any physical damage to any of the chromosomes, so they can get some information about the developing fetus at that time. That's typically why people might do it. So they get amniocentesis, and you have to collect these cells from the developing fetus. I asked my friend Terry once, he was getting amniocentesis done, to tell me about it. And Terry's kind of like a robot, so I knew I could ask. And most people, you don't want to talk about it until they say, oh, everything's fine. So I said, Terry, any word on the amnio outcome yet? He said, Jay, 10 days for results. The process was cool. 2 people do the amnio. One operates the ultrasound, the second the needle. The baby's head is about 1.5 inches in diameter now. She is moving a ton, kicking both legs out, raising her hand over her head, as in, don't stick me. They aimed for a space about the size of the baby's head in the sac above the head. They rammed the needle in without anesthetic and in one motion. The needle hit the small pocket perfectly, missing the head by about 3/4 of an inch. They then had to maneuver the needle through a membrane to get the good fluid with baby cells. They claimed to extract 1 ounce of fluid, but it looked like 4 to me. I wasn't sure if the needle ever grazed the skull. If not, it was within a few millimeters. 3 minutes, start to finish. So that's pretty much how the process goes. In a clinical way, there's Terry. There was the baby, Charlotte, and Terry's wife, Barbara. They can look at them on a computer. So you'll attach a microscope, put the cells under the microscope, attach a video camera, look at them just to see how many are there, and so on. Here's what they would look like just in the cell. This one has arrows next to 3 of them because it turns out this cell comes from an individual that has 3 copies of chromosome number 21. A cell expert would be able to see that and identify that. The genetic material then, these linear strands, that's where you're going to have the what Mendel called hereditary factors, the information. How do you make the enzyme that breaks down lactose? On chromosome number 14, you have the gene for milk digestion. But you have 2 copies, the maternal copy and the paternal copy. Then there'd be another gene somewhere else. The whole chromosome isn't filled with genes, but there are a lot of them on it. So you'll see in a genome, in my genome, I've got 2 copies of the gene for milk digestion, 1 maternal copy, 1 paternal copy. So Mendel, who's famous for 2 laws, but really it's the first law that is at the core of what is heredity, called his law of segregation, says this, that a gamete being a sperm or an egg, receives only 1 of the allele for every gene that the organism has. So you have your maternal copy and your paternal copy, but when you make a sperm or an egg, it only gets 1. And just like a lottery, whether it's this one or this one, the maternal or the paternal copy. So as you're producing sperm, if you're male, you're producing them, you have 2 copies of everything. Well, sometimes they'll get 1 of the alleles that you carry, sometimes they'll get the other. And for all of the 20,000 genes that you have as a human, sometimes they'll get 1, sometimes they'll get the other. So your sperm carries a mixture of your mother and father's genetic information that they gave to you. Same thing if you are an egg. Here's an egg moving down the oviduct. Same thing. Gets a maternal or a paternal version from the woman who produced the egg. And the last part of Mendel's law of segregation, it says, so you get only 1 of the 2 alleles that the individual has, but then fertilization reestablishes the diploid number. So you're haploid. You have 1 copy of everything in the sperm or egg, but then you get 2 at fertilization. That's it. That's what his law is. You start with 2, but then you make a sperm or egg that only has 1, and then 2 people put those together, and now the new offspring has 2. But the implications of that are pretty huge. So here's fertilization, risque slide. In Mendel's law of segregation, when you think about it then, it has some implications that are provocative, I would say. So we can look at now here, let's say we're talking about an albino giraffe. Every now and then you see them. They have a gene that both copies code for a non-functioning version of the gene that makes pigment. So if you had an albino, it would have 2 copies of the little m. A typically pigmented giraffe would have both copies of the dominant. When they produce the gametes, they have a little m. That's Mendel's law of segregation, right? Now, I've got the male producing sperm. They all have the little m. The eggs, they all have the big M. Here's what they look like. You don't see too many in nature. Makes them stand out even more than they already do at 14 feet tall. When those come together, if you've got big M eggs and little m sperm, every individual is going to be now diploid, the new individual, but they're going to have 1 little and 1 big. Because the big one masks the effect of the little one, they're all going to be normally pigmented. So their genotype would be little m, big M. This is the end of Mendel's law of segregation, that we've restored the diploid number, and we would call the phenotype normally pigmented. Now that individual, Mendel's law of segregation tells us, will produce half of their gametes will be a little m, and half will be a big M. At fertilization, you don't know which one is going to be involved in that. So if we were to cross 2 individuals that were normally pigmented, but they were heterozygous, then we're not sure what we're going to get. You'd have to look at them. So for that, we use our trusty Punnett square. named after Reginald Punnett, just a handy bookkeeping tool. And what you do is you fill in for the female. You put female gametes on top, typically. So all of her eggs are either a big M or a little m. The father is also heterozygous, so all of his sperm are either a big M or a little m. We don't know which one of the eggs is going to be fertilized. We don't know which one of the sperm is going to do the fertilizing. If it's that pair, the egg on top and the sperm on top, it would be big M, big M. Or if it's that sperm and that egg, it would be that, and so on. And so you fill in all of these looking at for a given sperm, move it over, for an egg, move it over. And now you see you've got all these options. And from that, you can deduce, all right, what are the genotype ratios? Oops, we had big M, big M, little m. So 1 out of the 4 was homozygous dominant. 2 out of 4 were heterozygous. And 1 out of 4 was homozygous recessive. In other words, genetics can tell you how the heredity works, but it still doesn't tell you which offspring you're going to get. It just says, on average, this is what it's going to be. We don't know because there is some randomness to the process. Mendel did this with peas, and it doesn't matter. Could have been cats or dogs or giraffes or tigers or humans, but peas are good because you can control which parent fertilizes which other individual. They have a pretty short generation time. You can produce tons of offspring from one cross. So they're a smart organism to use if you want to study heredity. Just it's faster and cheaper than using many mammalian types of species. The essence of his law of segregation, again, just to reiterate, because it's the most important thing in genetics, is gamete gets only one allele from the 2 that an individual has, and at fertilization, the diploid number is reestablished. Now, I said that there were provocative implications for this, and here's one of them. We talk about blood relatives. Everybody knows that phrase. You've got books called Blood Relatives, or you've got TV shows called Blood Relatives, and we just think in those terms. Well, that's because we imagine, initially, people thought, oh, there's something in our blood. That's what causes people to be the same. Wrong. It made sense, except that there is no blood being transferred from the father to the kid or from the mother to the kid. So it was an idea that didn't work. It, though, conveys how people just didn't know about those hereditary factors, the genes that were being passed on. Or we have this other idea that was popular from the 1600s all the way into the 1800s. It said, oh, every sperm has a tiny little human, a homunculus of a adult that then is going to grow. Well, that's the dumbest idea, right? You don't have to have a microscope or even Mendel to know that that's a non-starter, right? Right? You should be able to figure out why it is that that's a terrible idea for how heredity could work. So I'm not going to tell you the answer because you've had 200 years to figure it out. All right, I'll stop here, though. Mendel's law of segregation.